Hey guys, today we are starting the fourth and final unit of Bio 30, which is all about ecosystems and populations and that sort of thing. So today we're going to be getting into the Hardy-Weinberg principle. So the Hardy-Weinberg principle is just kind of a way to describe how a gene pool is changing over time. So we'll start with some definitions of gene pool and population. Then we'll talk about the different types of gene frequencies. And then we'll talk about what Hardy-Weinberg equilibrium is, the criteria to meet that equilibrium, and then we're going to finish off with some calculations involving the Hardy-Weinberg equation. So a population is a group of organisms that is in the same area at the same time and that is all the same species. In population genetics, we study how genes vary within a population and how both environmental factors and genetic factors affect a population. The variation makes up the gene pool of a population. So when we say a gene pool of a population, we're referring to all of the genes and all of the different alleles within that population. Gene pools do change from one generation to the next because some traits are selected for and become more common, and other traits are selected against and become less common. You also have mutations that occur that can introduce new genes or new alleles into the population. When there is breeding between different populations, that can also introduce new genes and new alleles. If populations are only bred amongst themselves, then you have to get like natural selection to be a force of change or mutations. Okay, so there are three different types of frequencies that we can measure when we're determining the variation of genes within a population or looking at how the gene pool is changing. The more variation in the gene pool, the more adaptable that population is and the less likely it is to go extinct. So that's why it's really important to understand these gene frequencies. So the first frequency is the genotype frequency, and that is the proportion of the population with different genotypes. So for instance, homozygous dominant, heterozygous, and homozygous recessive. So what is the frequency of those three different genotypes? Phenotype frequency refers to the population, the proportion of a population with a specific phenotype or trait. So usually how much of the population has the dominant trait and how much has the recessive trait. And then the allele frequency is the rate at which a certain allele occurs within a population. So the recessive allele or the dominant allele. To calculate genotype frequency, you just take the number of individuals for a genotype divided by the total number of individuals. To calculate phenotype frequency, you take the number of individuals with a specific trait and divide it by the total. And then to calculate allele frequency, you count the number of either dominant or recessive alleles and then divide it by the total number of alleles, which will be twice the number of people because every person or every individual has two alleles for each gene. And then all of the frequencies can be expressed as a decimal or a percentage if you just multiply by 100. Okay, so this is kind of a more simple example, but it says black fur is dominant to white. In a population of 5 individuals, you have these genotypes. So the genotype frequency would be 20% big B, because that's 1 in 5, 60% heterozygous, because that is 1, 2, 3 of 5, and then 20% homozygous recessive, because again, that is 1 of 5. And then for phenotype frequency, well, out of these 5 genotypes, 4 of them, the first 4, all code for black fur because they all have that dominant big B allele. So 80% of these individuals will have black fur, and only 20%, the little b, will have the white fur. And then the allele frequency would be 50% B, big B, 50% little b. And that's just if you count how many big B's and how many little b's there are in this group here, there's equal numbers of both. Now, the important part to note here is that you cannot determine the phenotype frequency of the dominant trait by only knowing either the homozygous dominant or the heterozygous genotype. But the homozygous recessive genotype frequency will always match the recessive phenotype frequency. So these numbers will always be the same as each other. This number and this number will not be the same, and this number and this number will not be the same. Yeah, I'm actually going to skip this because even though I've introduced gene frequency questions like this, usually we're going to be solving all of these gene frequencies, not just by counting alleles like this, but by actually using the Hardy-Weinberg equation. So we're just going to go to that instead. So Hardy-Weinberg equilibrium is a proposal by two scientists, Hardy and Weinberg, about how to determine genotype and allele frequency. Essentially, they said that the genotype and allele frequencies of a population would not change from one generation to the next if the population is at equilibrium. And there are 5 conditions that a population has to meet in order to be at equilibrium. Most populations don't actually satisfy all 5 of these criteria. They might meet a couple of them, but it's rare for a population to meet all of them. And so most populations are not at equilibrium, which is why most populations have changing gene frequencies. But we're going to look at it anyway. So the first condition that populations need to meet in order to be at equilibrium is that they have to have a large breeding population. So essentially, there has to be a lot of individuals contributing to the gene pool for the next generation. If you have a really small breeding pool, it's just like if you have too small of a sample size in like a survey or an experiment. There can be just random changes from what you would expect. So for instance, if I flip a coin twice and get heads both times, I might say, oh, if you flip a coin, you get heads 100% of the time. But that's not accurate. If I flipped a coin 1,000 times, I would get roughly a 50-50 split between heads and tails. The smaller your sample size, the less accurate your predictions can be about statistics and stuff like that. So if you have a really small breeding population, the gene frequencies and allele frequencies can change just based on random chance, the way you could, through random chance, flip a head twice in a row. So you have to keep a large breeding population, so you have a large sample size, and you don't have those like random statistical anomalies. Yeah, large sample populations are more stable, and therefore the next generation's genes are more likely to follow a predictable pattern. The second condition is random mating, which means that there can't be any sexual selection. As soon as like a male bumps into a female, they'll mate with each other. There's no like needing to be attracted or something like that. So because the mating is random, it means that there will be no sexual selection of certain traits. Therefore, those traits don't become more common over time. So if there was sexual selection, that would change the gene pool of the next generation because desirable traits would become more common. The third condition is that there can't be any mutations that would randomly input new alleles or new genes into the population. The fourth condition is that there can't be any gene flow. What that means is there can't be any movement of genes between different populations. There can't be any emigration, no emigration, no individuals from population A mating with individuals from population B. All of the mating has to stay within the population and no one's allowed to come or go. The reason for that is that as soon as you have the movement of individuals or genes between populations, that is going to introduce new genes and new alleles to a population. So the population has to be reproductively isolated, is another way to say that there can't be any gene flow. And then lastly, there can't be any natural selection. If a trait increases the survival or reproductive success of an individual, it will be selected for and it will become more common in the next generation. So if we want the gene frequencies to stay the same from one generation to the next, which is what equilibrium is, then there can't be any selection of certain traits because that would increase their frequency. So just like there can't be any sexual selection and the mating has to be random, there can't be any natural selection either, because both of those things would change the frequency of genes in the next generation. So like I said, lots of populations can meet some of these criteria, but it's difficult to meet all 5. Large breeding population is really easy to meet. But mating is not random in quite a few species. It's almost impossible to say that there's not going to be any mutations. It's very difficult to have the no gene flow unless you have a population like alone on a distant island or something like that. And natural selection happens with most species unless there is no competition, no death, nothing, no predators. Tons of food, tons of water, tons of mates. Unless all of that's true, there's going to be something that makes certain traits better than others. As soon as some individuals are dying or some individuals are out competing others, then you're going to have natural selection. So because of that, gene frequencies and populations do normally change. So the Hardy-Weinberg principle essentially summarizes the way we can calculate genotype frequencies and phenotype frequencies and allele frequencies and stuff like that within a population. We can compare what we expect the frequencies to be based on these calculations to the actual frequencies to see how much the gene frequencies are changing within a population. That lets us know how healthy the population is, how adaptable it is, how vulnerable to extinction it is, that kind of thing. So these are the 2 Hardy-Weinberg equations that make up the Hardy-Weinberg principle. The first one describes allele frequency, so P plus Q equals 1. And then the second one describes genotype frequency, P squared plus 2PQ plus Q squared equals 1. So P and Q are the allele frequencies. P is the dominant allele, Q is the recessive allele, and then P squared is the homozygous dominant genotype, Q squared is the frequency for homozygous recessive, and 2PQ is the heterozygous genotype. So when we're doing different Hardy-Weinberg problems, the first thing you want to identify is it talking about allele frequency or genotype frequency? Because those are 2 completely different equations. So yeah, just make sure that you're very clear on which of those it's asking for. Okay, we can walk through how I did this. So anytime you're given phenotype ratios, which is what this is, 60% red, 40% blue, red and blue are traits, so that is a phenotype ratio. And then it says red is dominant, it wants the allele and the genotype ratios. So the kind of starting point anytime you have phenotype ratios is always identify your recessive phenotype, because the recessive phenotype ratio is equal to the recessive genotype ratio, homozygous recessive genotype. So that's what's happening over here. I'm saying that blue, which is our recessive in this case, is going to be little r, so that's 0.4. The 0.6 red, I don't know if they're homozygous dominant or heterozygous. That's why that 0.6 isn't useful. But the 0.4 is definitely homozygous recessive. So I can say that 0.4 is equal to r squared, or q squared if you want to use your p's and q's. So I'm just going to square root that to figure out what my r value is. So if I square root 0.4, I get 0.632. Then I can plug that into my allele frequency formula to solve for the frequency of my P allele, which is my dominant allele, big R. Then once I have both my P, which is my big R, and my Q value, which is my little r, I can plug it into the other Hardy-Weinberg equation, P squared plus 2PQ plus Q squared equals 1, and then I just solve from there. I don't actually add these together. You can add them together to check, and they should be roughly 1, but you actually keep these separate. And if it was only asking what is the genotype ratio for heterozygous, you would only have to do the middle part of this, just the 2PQ. If it asked just for the percent of the population that was homozygous dominant, you'd only have to do the P squared part of this. Okay, but because it asked for all three, I can say that 0.135 or 13.5% are homozygous dominant, 0.456 or 46.6% are homozygous, that should be 45.6, sorry, 45.6 are heterozygous, and 0.4 or 40% are homozygous recessive. Okay, so let's walk through, yeah, let's do the first problem together, and then you guys can try the next two on your own. Okay, so for the first question, it tells us that H is 0.2. And because this is a lowercase letter, what that's really telling us is that Q is equal to 0.2. And it wants to know the genotype ratio. So I'm going to start with my formula P plus Q equals 1, which then tells me that P plus 0.2 equals 1. And if I move the 0.2 to the other side, I get a P value of 0.8. And then I can use the second formula, P squared plus 2PQ plus Q squared, to find my genotype ratio. So P squared is going to be 0.8 squared plus 2 times 0.8 times 0.2 plus 0.2 squared. And again, it should all equal 1, but we don't actually add it together or do anything with that. So 0.8 squared is 0.64. Then plus 0.32 plus 0.04. So that means that I've got 64% that would be big H, 32% that are heterozygous, and 4% that are homozygous recessive. That would be my genotype ratio. Okay, so try number 2 and 3 on your own and then come back for the answers. Okay, so it says 70% of the population has the dominant trait, which is green. Again, the dominant trait could be 2 different genotypes. So the dominant trait on its own isn't really useful to me. What is useful to me is knowing what % of the population has the recessive trait. And so in order to figure that out, I just do 100% minus the 70% that are dominant. The remaining 30% must be recessive. So I'm just going to turn that into a decimal. So that means 0.3% have the recessive trait, which is, in this case, little g, little g. Now, the recessive genotype, in terms of Hardy-Weinberg equation, is q squared. So that means that 0.3 equals q squared. I then want to figure out what just q is. So I'm going to square root that to get just q. And that is 0.5477-ish. Now that I have q, I can figure out what p is. So 1 minus 0.5477 is 0.4523. So my allele frequency, as far as P to Q, is roughly 45% to 55%. About 45% P and about 55% Q. Then it wants the genotype frequency, which is where our P squared plus 2PQ plus Q squared comes in. So I'll do 0.4523 squared plus 2 times 0.4523 times 0.477 plus 0.5477 squared. So that'll get roughly 0.2 for my P squared. 2 times 0.452. And 0.495. And 0.3. Okay. So I'm going to round that to 20%, 50%, and 30%, so that it adds to 100%, or so our decimals add to 1. And that is going to be my genotype ratio of homozygous dominant to heterozygous to homozygous recessive. Okay. So step 1, always figure out how much of your recessive trait you have. From that, figure out what your Q is. Then figure out what your P is using P plus Q equals 1. And then lastly, plug it into the bigger formula to figure out your genotype frequency. So that's what it all looks like in the end. Okay, let's try the last one. It says that 1 in 25 have the recessive trait. So 1 divided by 25 is 0.04, and a recessive trait is our Q squared. So we'll start by figuring out that 4% of our population are 0.04, are Q squared. Then I square root it to get 0.2, and that is going to be the amount of individuals in the population that carry the recessive allele. So that's our Q, is our allele frequency. Then I can find my P. It's going to be 1 minus the 0.2, so 0.8 of the population is going to carry the dominant allele. Then I can go to my P squared plus 2PQ plus Q squared. 0.8 squared is 0.64. 2 times 0.8 times 0.2 is 0.32. And then 2 squared is 0.04. So we've got 64% that are going to be homozygous dominant, whatever letter that is. 32%, which would be heterozygous, and 4% that would be recessive, homozygous recessive. Okay? So just remember, anytime you're seeing Q squareds, that is always your recessive trait, but also your recessive, homozygous recessive genotype, which we can use to get our allele frequency for Q, which we can then use to get our allele frequency for P. So these are allele. Anytime you have just the P or just the Q, and then these are our genotype frequencies. Okay, I will be giving you guys more practice with Hardy-Weinberg equations and stuff like that, but let me know if you have any questions.